Hello everyone, I welcome you all to another session from Biotechnica, and today we are going to discuss about cellular signaling mechanisms, that is a detailed study about the components that are involved in signaling. And this is considered to be one of the important topic when I speak of CSIR NET Life Science exam. So let's start with the session. So what is cellular signaling? Well, as you all know, it is a process by which cells detect and respond to the signals in their environment. So what kind of signals it will be? It could be in the form of hormones, growth factors, cytokines, neurotransmitters, or any kind of environmental signals. So obviously if signals are there, corresponding to that receptors will be there. So those receptors can be present either on the cell surface or maybe within the cells. And the nature of receptors will be proteinaceous by nature. Now, looking into the types of signaling pathways. So we can classify them as autocrine, paracrine, endocrine, juxtacrine, and synaptic signaling. So among the signaling pathways, so if we compare autocrine, endocrine and paracrine, endocrine signaling is all about long distance signaling because it travels through the bloodstream to reach the target cell. Whereas paracrine signaling is a short distance signaling because if suppose this is a cell A and this is cell B, cell A releases certain chemicals that will have its effect only on the B, that means nearby cells. Autocrine signaling, it is something like the A cell produces the chemicals, but it will be having its effect on itself only, not on the neighboring cells. So I have another image to explain you the remaining signaling pathways. Contact dependent is nothing but a juxtacrine signaling pathway. That means where a signaling cell and the target cell should be in close proximity. Then only the signaling happens. As I told you, paracrine means local mediators. The target cells will be nearby. Similarly, endocrine signaling means they will be passing through the bloodstream and to reach the target cell to carry out their mode of action. On the other hand, synaptic signaling means it is related to neuromuscular junction or neuronal transmission, wherein the presynaptic neurons will be having the neurotransmitters present and the target cell will be having the corresponding receptors. So in between the two cells, that is the presynaptic neuron and the target cell, we have the synaptic junction present. So when these neurotransmitters are released into the synaptic junction, attaches to the target cell, thereby inducing the neuronal transmission. So this is about the basic types of signaling mechanism. Now, what are the molecular mechanism that is involved with signal transduction? So if you can see, we have 4 important features for every signal transduction pathway. First one is specificity. That means a particular signaling molecule will have its affinity towards their corresponding receptor. Similarly, if another molecule comes and tries to bind to this receptor, it is not possible. Why? Because they lack the complementarity. For example, we have insulin. It will go and bind to the insulin receptor only. It will not go and bind to the glucagon receptor. Why? Because of lack of complementarity. Second, once a signal goes and binds to the corresponding receptors, downstream proteins will be activated. So it is not a single molecule getting activated, but a group of or a collection of molecules are getting activated. Now that could be in the form of proteins or enzymes. So that is known as amplification. That is, the number of affected molecules increases geometrically in an enzymatic cascade. Third important mechanism is desensitization or adaptation. That means when a signal goes and binds to the corresponding receptor up to a particular period of time or after a particular period of time, it will not generate any response. The response itself will give a negative feedback. So this is basically a kind of termination process or one of the method of terminating a signal transduction pathway. And the last one is integration. Integration happens sometimes. That is, when two different signals goes and binds to their corresponding receptors, they combine together generating a common response. That is known as integrating or combining the two different pathways leading to a common response. Next, as I mentioned in the beginning, we have two types of receptors present. So based on that, we can classify it as hydrophobic signals and hydrophilic signals. Hydrophobic signals like steroids, retinoids and thyroxine will be having cytosolic receptor. Upon binding, they will migrate into the nucleus to carry out the gene expression. Whereas hydrophilic signals like small molecules, peptides and proteins, they have cell surface receptors. Once bound with the ligand, they will activate certain intermediate proteins and moves into the nucleus to carry out the gene expression. Next, we have the components that are involved in signaling mechanism. Obviously, every signaling begins when a ligand goes and binds to the receptor. So, receptor can be cytosolic or cell surface. Because of this binding, it is going to activate intracellular signaling proteins. Now, that includes effector proteins, second messengers, protein kinases. And these components will be varying depending upon the signaling pathway. So, you can create a comparison table and study that will be easy for revision. Then, once these kinases are activated, obviously the function of kinase is to add phosphate to the target and they will generate the cellular response. Or any kind of cellular response. It could be altering a metabolism or maybe the gene expression or maybe the movement, cell shape or movement. That is, if it is related to cytoskeletal protein. And finally, the termination process happens. Right? So, I have given you an insight regarding the signal transduction pathway. What are the components? What are the types? Now, let's go into more elaboration from the examination point of view. So, these are the major signaling pathways, and among which, let me say you few signaling pathways that are crucial or important from examination point of view. That includes receptor enzymes, serpentine receptors, which is commonly known as GPCR, as well as receptor with no intrinsic enzyme activity. So, we have all total 6 signaling pathways, but the prime focus will be on these 3 signaling pathways. To begin with, we have gated ion channels. As the name indicates, when a ligand goes and binds to the receptor, it will allow the opening of ion channels. The gate will be opened and this will come inside, the ions will come inside, thereby changing the membrane potential. Receptor enzymes, as the name indicates, the receptor itself is having the enzymatic activity. Well, it could be a tyrosine kinase or serine threonine kinase. So, when the ligand goes and binds to the receptor, this kinase activity will be activated and it will carry out or activate the downstream proteins. Third, we are having receptor without intrinsic enzyme activity. Now, what does it mean? When a ligand goes and binds to the receptor is not having the enzymatic activity. So, we have cytosolic kinases, or we can say cytosolic tyrosine kinase, that will be recruited to the receptor, which will help in the autophosphorylation event and finally leading to the activation of downstream proteins, thereby generating the cellular response. Next we have the fourth one, the most important one, that is serpentine receptors. We commonly call it as a GPCR, 7 transmembrane receptors, wherein the ligand will go and bind to the receptor, activates the G proteins, corresponding effector proteins and second messengers will be activated, leading to cellular response. Fifth one is steroid receptors. So basically these are nuclear receptors, and the ligand will go and bind to the receptor directly, thereby regulating the gene expression. And the last one is the less important or the least important one is the adhesion receptor. Now this is related to ECM proteins, wherein the ECM proteins will serve as the ligand and goes and binds to the cell surface receptor, that is integrin, and how they are generating the response. So in this session, I'll be focusing on these 3 pathways, that is receptor enzymes, GPCR, and receptor without enzymatic activity. So the first one is G protein coupled receptors. As I told you, it's a 7 transmembrane helices having extracellular segment that is responsible for glycosylation, transmembrane region for hydrophobic amino acids, and cytosolic region where the G protein can go and bind, specifically between C3 and C4. Next, G protein is a family of membrane proteins that are anchored to the membrane. They will recognize activated GPCR and pass their message to the effector system. It is named as G protein because of their interaction with GTP as well as GDP. And it is composed of 3 subunits, alpha, beta and gamma. Among the 3 subunits, alpha is having the GTPase enzymatic activity. Thus, G protein function as molecular switch because when they are bound with GTP, they are on, and when they are bound with GDP, they are off. So to explain this concept, I have an image for you. See, this is the resting stage wherein the receptor is inactive, G protein is there in the form of trimeric, and this is your effector protein in the inactive conformation. So when a hormone goes and binds to the receptor is activated, which in turn undergoes conformational change. As you can see in the difference in the structure of the receptor. Now this will allow or open the binding site for G protein. When this G protein goes and binds to the receptor, GDP will be released, GTP will go inside. So when GTP is bound, the alpha, beta, gamma subunits will be separated. So this G alpha GTP will go and bind to the effector proteins, thereby activating the effector and producing the signal transduction mechanism. So if you want to terminate this pathway, we have different mechanisms for that. For example, one way is hydrolysis of GTP. Once it is done, it comes back to the resting stage. Other ways, phosphodiesterase can be used. Now that will help in the breakdown of second messengers. Or as I mentioned in the previous slide, desensitization. So any of this method can be used for termination purpose. So these are different types of G protein. We have G alpha S, alpha I, alpha Q and alpha T to be focused on. But in this session, I'll be taking alpha S and alpha T, which is the most important among the 4. So G alpha or GS pathway, it's very simple if you know the components. See, epinephrine, when bound to the corresponding receptor, there will be an exchange of GDP to GTP. So beta and gamma will be separated. The GS alpha combines with the GTP, activating the effector protein adenylyl cyclase. This enzyme catalyzes the conversion of ATP to the second messenger, that is CAMP, which in turn activates the protein kinase A, thereby phosphorylating the target proteins to generate the cellular response. in case if you are adding phosphodiesterase here, it will result in the breakdown of second messengers. So this will result in termination process. So next pathway is GT pathway. This is also important, as many a times, even recently also a question came from here, and most of the times you will find that they are being asked in 4 marks, part C question. So you can separate this GT pathways into 3 phases. One is the excitation phase, that is the light phase. Next you have got the dark phase, and next you have got the recovery phase. So rhodopsin is the protein that receives the light signal. So upon absorbing the light, the 7,11-cis retinal present in the rhodopsin protein gets converted to all trans retinal. So this results in rhodopsin activation, which in turn activates a G protein known as transducin. As usual, there will be an exchange of GDP to GTP on transducin protein. So once the protein is activated, it will remove the inhibitory subunit from phosphodiesterase, which in turn activates the phosphodiesterase. So once the phosphodiesterase is activated, it is responsible for breakdown of second messengers. So what happens? The ion channels, that is CGMP specific ion channels, will be closed. See, the cation channels will be closed. Influx of sodium and calcium will be prevented. Thereby, the membrane becomes hyperpolarized. So this signal goes to the brain. And because of continuous efflux of calcium ions through sodium-calcium exchanger, there will be reduced concentration of cytosolic calcium ions in them. So this is what happens in the light phase. So when we enter into the dark phase, what happens? We have already the low calcium ions. Now what is the benefit of this low calcium ions? On one hand, it will be activating guanylyl cyclase. On the other hand, it will be inhibiting phosphodiesterase because it should not break the second messengers. GC in turn will activate the second messenger, that is CGMP. And CGMP will activate the protein kinase G, which will activate the CGMP specific ion channels. So what happens? It results in reopening of cation channels and returning back to the pre-stimulus level. So that completes the dark phase. And the last one is the recovery or the adaptation phase. What happens here? The rhodopsin protein will be phosphorylated by rhodopsin kinase. And this will be stabilized by another protein known as recoverin. Phosphorylated rhodopsin is activated, or it is attracted by another protein known as arrestin. After some time, what happens? Arrestin will be dissociated. And as you can see in the image, the all trans retinal is going out, 11-cis retinal is coming in. So this is bringing the rhodopsin protein back to the initial position. Now that is ready to take up another phototransduction cycle. So remember, from this pathway you can expect either in the form of match the following or statement based questions. So it is very important to remember all the 3 phases, the sequence of events that occurs in each phase. Next, if we can see receptor enzymes, it is classified into 3 major types. Tyrosine kinase pathway, under which we have RAS, PI3K and FOXO1 degradation pathway. Second one is serine threonine kinase pathway, for which TGF-beta is the example. And third one is the GC pathway or guanylyl cyclase pathway. Well, this is not basically a pathway. It is just they are mentioning which enzyme or which protein is present in which location, what is their function. That's it. So it can be classified as membrane-bound GC and soluble GC. So based on that, we have different types of ligands. But from examination point of view, the most important pathway is RAS pathway. So let's have a look on to that. So RAS pathway, all it begins with insulin as a ligand when it goes and binds to the corresponding receptor. The receptor is heterodimer. Why? Because it is having 2 alpha and 2 beta subunits. It is having the tyrosine kinase activity. So when this insulin goes and binds to the alpha subunit of the receptor, the beta subunit undergoes autophosphorylation. Now this will recruit the substrate to the receptor, that is insulin receptor substrate 1. So they will be interacting with each other via PTB domain. So PTB domain is having affinity towards phosphorylated tyrosine kinase residue. Next downstream protein is GRB2. GRB2 serves as an adapter protein. Why I called it as an adapter protein? Because it interacts with IRS1 via SH2 domain and interacts with SOS via SH3 domain. SH3 is proline-rich domain. The next protein over here is SOS, and SOS is considered to be a GEF, that is guanine exchange factor. As you can see on the RAS protein, GDP is going out and GTP is coming in, so thereby activating the RAS protein. This in turn activates the RAF1 protein. RAF1 in turn phosphorylates the MEC protein at 2 serine residues, thereby activating it. This MEC in turn phosphorylates 2 ERK protein, phosphorylates 1 threonine and 1 tyrosine residue on ERK protein, thereby activating it. So once ERK is activated, it will be released into the nucleus, combines with the transcription factors like SRF and ELK1, and they carry out the gene transcription necessary for cell division. So based upon the discovery, RAF1 is named as MAP, kinase, MEC, MAP kinase, and ERK, which was first discovered belonging to the family of MAP cascades. So it is termed as MAP kinase. And the last pathway to be discussed is JAK-STAT pathway, or the receptor without enzymatic activity. So here we can also say that two different pathways can combine together, giving rise to a single response. So let's take the example of a cytokine, that is erythropoietin. So this erythropoietin goes and binds to the corresponding receptor. As I said, it is a receptor with no intrinsic activity. So we have soluble cytosolic tyrosine kinase being recruited to the receptor, and the example for that is JAK. This phosphorylate, so they will undergo autophosphorylation and they will attract a downstream protein known as STAT. So they will be having the SH2 domain, so via SH2 domain they can interact with each other. After that, the STAT will undergo dimerization so that NLS can be exposed. Upon dimerization, they will transphosphorylate each other. Once the NLS is exposed, they can enter inside the nucleus, carry out the gene expression. Otherwise, since it is having phosphorylated tyrosine residue, it is also having the affinity for binding to the GRB2, which I had mentioned previously. So they will activate the MAP cascade, thereby entering inside the nucleus, carrying out the same gene expression. So these are some of the important pathways to be focused on when you go for cell signaling. Apart from that, other pathways are important, but these are the important pathways. You can drop a comment for any signaling pathways if you want to know in more elaboration. Thank you so much.